This video will focus on the effects of different conditions on fungal life processes. So we know that there are a few different factors that can affect the process, the life processes, being temperature, oxygen, water, and pH. Different organisms react to their environment in different ways. So we know that they have adaptations to the environment they live in. Any environment or condition that changes may actually affect the organism itself. So some microbes can remain dormant until conditions are favorable. So if conditions, for example, there's too much oxygen present or the temperature is too high, some organisms will encapsulate themselves for protection and stay dormant until the conditions are right, like the oxygen is decreased or the temperature is decreased, and they can actually start reproducing. So let's look at temperature first of all. Now, if we can see here in this graph, we've got the volume of gas produced in hours, and we have temperature on the x-axis. We can see that the temperature increases up until around 40 degrees Celsius, and then there is a decrease in the volume of gas. So basically the ideal temperature is close to 45 degrees Celsius. This is when the cells are respiring the most, and we know that carbon dioxide is produced with aerobic respiration, so we can actually measure the carbon dioxide present or the gas produced. Okay, enzymes control that reaction, so enzymes need to operate at certain conditions, otherwise they will denature. The optimum temperature is 45 degrees in this case. The next being oxygen. So we know that there's aerobic and anaerobic organisms. Some organisms live in different environments, and that can actually really affect their respiration rates or their life processes. So we can see here aerobic respiration in bacteria, sugar and oxygen produces carbon dioxide and water. Aerobic respiration in fungi is exactly the same. Anaerobic respiration in bacteria, lactic acid is produced. Anaerobic respiration in fungi, alcohol and carbon dioxide is produced. So those are the differences there. The next is water. So organisms do require water just like any other organism does, and it's a crucial factor for growth. Now, some term that you may have come across before is osmosis. Now, this is known as the movement of water from an area of high concentration to low concentration through a semi-permeable membrane. Now, if you remember back to the fungi itself, the structure, we didn't really cover it, but it is does have a membrane surrounding it, just like a bacteria does, and just like most cells do. So the membrane itself is used for transporting substances. Some cases, in most cases, water is transported through here. And basically, there are a few different factors that can influence how much water is entering or exiting the cell. If microbes are placed in a low solute concentration, okay, say for example, you're placing a bacteria into a beaker of salt water, and the solute of salt, sorry, the concentration of salt in the beaker is lower than it is in the inside of the cell, the water will enter the cell. This is because it's moving from an area of high water concentration, which is outside the cell, to inside the cell. This is going to cause the cell to swell and to burst. On the other hand, if the microbe is placed in a high solute concentration, Okay, so the beaker, the salt water in the beaker is very salty, has a high concentration compared to the cell. The water will exit the cell, causing it to shrivel up and die. Okay, and that is it. So in the next video, we will focus on virus structure.